So welcome to video 6 in G02 on genome assembly, where we're looking at the practical and concepts behind it before we actually apply it in the demonstration video. So please look at those videos to understand how to actually apply the steps. We're going to talk about the background to it first. So it's ideal that you view this video before attempting the steps. So what we're going to do in the practical is, first of all, get our Illumina and Nanopore data. You can use existing files that are available on the server that I can point you towards, or you can get your own data. It's whatever you want to use, but I would advise getting a set of short reads and a set of long reads, so you can look at the differences in the outcomes, preferably for the same species or sample. We're going to de novo assemble those with spades. So spades can tolerate short and or long reads. It works well with these, and there's a variety of features that allow different types of assembly depending on what you're looking at. So it's quite actually applicable to viruses, bacteria, fungi, simple eukaryotes, and longer eukaryotic genomes as well. We're going to evaluate these outputs with QUAST, which is a standard evaluation tools in the same way that spades the standard de novo assembly tools, and then we'll visualize the data with Bandage. And finally, we'll aggregate this information from QUAST and Bandage to evaluate these assemblies. That's our goal for the practical. So what we're doing is bringing ourselves through from this initial stages where we're looking at QC, doing trimming and filtering and all the rest on our data, so that we could now assemble our sequence and then also look at the result to see did it actually work well. And because we're looking at multiple attempts of assemblies with different k-mers, that can be complicated. So another way of looking at it is in this graph that we had our sequencing data initially generated, that we did the QC part in the last practical. Here we're doing de novo assembly in G03. We'll do remapping, and then we'll look at a variant calling in G04. So that's how our courses link together. As you know from my last work, that irrespective of what sequencer we use in our sequencing source, that ultimately we get FASTQ files that we're working with. So initially they might be converted into different files that are base called, then to convert them into FASTQ. But that's mainly what we're dealing with. When we look at subsequent courses, we look at other FASTQ related file types known as SAM and BAM and BCF and VCF, but we won't be dealing with them directly in this course. What we're trying to do is tackle this kind of very complex map of different tools we can use and take our initial data here where we did some sort of quality control and then look at de novo assembly. There's different de novo assembly approaches depending on what you're trying to do, and we're looking at a more simple one. Whereas if you're looking at a more complex, maybe a plant genome, you might want to try different things. So we're looking at here de novo assembly, and we're going to touch on assembly finishing, at least in the theory sense. What we're dealing with this practical are mainly FASTQ files up to this stage, and we're going to generate FASTA files from the de novo assembly. An example workflow is available from the Arctic system for looking at SARS-CoV-2 sequence data. So you might be looking, say, at Illumina data initially, and you might have some sort of QC approach like trim galore, and then you might wrap the reads with BWA, and then use a sequence, another tool to get rid of primers, so you can call variants and look at that. Another approach that is more similar. What we're doing is we're taking, say, Nanopore data. We're done QC already. We'll use tools that will help do a de novo assembly without a reference in our case. And that will let us perform additional features later. And it's worth bearing in mind that, like all areas I alluded to in the G01 practical, is that it's a lot, what we're doing is a lot like cooking, that we're taking in initial data to create something from it. But what exactly we want to create depends on the context of what you're trying to do. We have algorithms and appliances that are available and particular recipes for going about this. And our recipes, in a sense, is our de Boer graph approach that we're mainly going to use here, where we're going to do that with multiple k-mers. And that will ideally give us a good assembly at the end, but you might find that your data does not actually assemble well. That might depend on our initial ingredients. If you have poor ingredients, it can be hard to get to the desired outcome at the end, even if we try our best. So that's kind of where we're at. We've these FASTQ files, and then we have our tools that apply the Boer graph approaches, and then we ideally get our assembly at the end. The main suggested data that we're dealing with is a SARS-CoV-2 genome, and it's worth noting that you should have a single assembly of about 30 kb, give or take. You might find that there's primer sequences or other things inside there that cause it to be longer than that. And you might find that there might be certain regions that fall out as individual contigs, say if there's low complexity regions at certain locations. You should ideally use the libraries that we have already, and we'll talk about in the practical the ones that you can use, which are in my assembly folder. So with the spades approach, you can look at your log file in the output when you run it. What it's doing basically is it's using by default a set of k-mer values to perform the de novo assembly. So those k-mers have different sizes, and these are the detailed options. It's probably best to go with the default for your assemblies unless you have something particularly peculiar. And with viral genome sequence data, you can use, longer, more unique k-mers, and you could provide it with a wider span of them because it will run very quickly. It might take maybe 4 minutes on your data, give or take, to run. So it is possible to give it more k-mers, particularly more unique ones, to see if that works better. It will correct these reads or the k-mers with base hammers, index these k-mers, and construct the initial graph and try to prune reads as it go along to take out erroneous ones that don't actually connect further. So I'll gradually get rid of those and reiterate that process to join the nodes together into contigs. Then at the end of this, it will look across the different k-mers and use that information to try to identify potential mismatches that are incorrect and merge these assemblies to create a consensus assembly across these k-mers. So the tool that we're going to use to download the SARS-CoV-2 reference or the reference that you want is called ncvi ack download, which you can install with pip. So you'll run this command to download that tool and then be able to run it as that tool name with a format that you want, like FASTA or GenBank, and the accession number that you want at the end. I know this is the exact version of the accession number that you're using, and that will download that FASTA file or GenBank file of interest to your local folder. What we'll do with cost then, we'll run it. So we'll be able to look at the sizes of the different contigs depending on the length. Obviously, with SARS-CoV-2, you won't get any that are much longer, but you might find that you get actually many contigs for certain types of data. You might find the total length is a certain value. And you'll be able to look at all the features like the largest contig, the GC content and that of the reference, and the key value metric like the N50 value. So those are all important for trying to understand our assembly. You'll also be able to look at the numbers of misassemblies. So ideally that should be low, and ideally we want few contigs that are misassembled. You'll be able to look at the redundancy among contigs, which is quite high here. Ideally you would want a value close to 1 instead of this very duplicate ratio, which indicates there's a lot of redundancy in the assembled contigs, which means that the assembly process didn't work very well. And you'll be able to look at other features like the numbers of indels and other metrics that really are related to the N50. When you look across, you'll be able to look at the contig size viewer, which will give you the different contigs shown here. This is the contig and this is another contig. You see that they have these regions in white here where there's a gap. And this is a zoomed in region 2.7 kb. So you can see that these contigs are quite short. We ideally want them to be bigger. And this is our full genome sequence down here with each of the contigs shown in red. So you can see that this is actually a poor assembly. You can see there's a lot of misassembled contigs. We'll also use Bandage, and that will visualize these contigs as a deformed graph. So you can see this is a node and it's connected by these black edges to these two small nodes here. So there's some sort of bifurcation here before it connects to this node here, and we have more nodes out here. So that is kind of a visualization tool. You can run it on the server or download it to your local computer. That will mean you can check the assembly contigs quality and do blasts on contigs that are maybe odd, like these ones here. So you can think of this that you're taking your initial assembly graph, which might look mixed up together, and trying to identify what is the true path here manually. So what we want, say if we're looking at a plasmid, will be something that's a single circular contig, whereas if we get many short contigs, that's obviously an indication that our reads have not been assembled in the manner we would like. It could also mean that the region we're looking at is highly, highly variable. And then what we can do at the end is evaluate assemblies for different k-mers or different k-mer sets and compare the key metrics like the coverage levels to see how that changes. And as you see here, as we have more uniqueness here, we get less read coverage, which is expected. We have fewer nodes as we increase the k-mer. The N50 increases, and that's really a key metric. So this would indicate that the assembly is getting better. The maximum N50 associated with the longest contig is improving too. On the total number of bases, well, it falls a bit, which again you would expect because erroneous reads are being removed. And you see there as well that the reads used also decreases. So the better assembly is actually using less reads overall. And then we can look at other things like the total contig length greater than 1 kb, and this is for a single cell eukaryote of about, its actual genome length is about 36 megabases. So this assembly is not very good, really. And we see the number of contigs with greater than 1 kb is quite large. This is the N50 value of 4.7 kb, which is okay for viruses, but not good for host or vector genomes. And the k-mer coverage is somewhat low here. So what we're looking at when we're looking at the N50 is the minimum length among the contigs that span 50% at least of the genome. So basically we take our contigs down here, we sort them and determine the midpoint where they cover half of the total assembled sequence. So if this one, let's say, is 100 bases, and the next one here is 90 bases, and the next one here is 80 bases, and so on, our N50 value is going to be 90. So that's reflected in our stats here. That's what we actually have for this example. So what we do with our assembly, if we have contigs for host and vector genomes, that we convert our contigs into scaffolds, and these scaffolds into chromosomes. And what we want to do then is use the best reference we can access to align, order, and orient these contigs into scaffolds. So we compare them to a reference, we order them in right order. If they have gaps represented by ends between them, then we order them so they're in right orientation, so that we have our initial contig A here, and we have a gap until B, that we can at least estimate the likely gaps between them, and at least have them in the right order using a known reference genome, which is what you might do with this kind of data here, which is not great. This process is known as configuration. So it's this combination of procedures, alignment, ordering, and orienting. So this requires a known genome. So if you don't have a genome map, that's more challenging. So we then move on to look at the N50 metrics for these continuated contigs. And you can look at how they work well. So let's say we have another example here that this is 90, and this one is 70, and this one is 60, and then we have 40, and so on. That would mean that the N50 here is 70, because that's the length of the contig that passes this midpoint here. If we have a poor assembly, though, that can create issues because we actually falsely determine the midpoint to be much more shorter, and we might determine that the N50 here is actually longer because it has an N50 of 100, even though the other contigs, let's say of base lengths 20, are not very good. So we also need to look at the total span, which you saw earlier, the total bases, to see if that's consistent. And we had other examples here the total length, if that's out of sync with the reference, like this example here, that's indication that this assembly is not very good. And we shouldn't be really trying to, the M50 stat might be biased. So let's look at this M50 and how we can calculate it. So let's say we have 7 contacts with these lengths. What you can see is that this is already sorted. So you want to add all these up. So if we add these up, we have 15, 22, 30, 41, 56. So if we have a sum of 56, what we're going to be looking for is the point at which the base 28 is passed. So that means we have to go 26 and 34. So it's going to be an N50 value we get for this is 8, because that sums to 34. In the same way that we applied that to these ones. So we say we have 6 contacts. We perform the same procedure. So we just add up the numbers. We have 19, 30, 42, 57 for this. So again, we're looking at the point at which it passes halfway, which again is going to be 27.5, is it? No. Sorry, 28.5. So that's 27 for these two. So again, this is going to be 11 when it passes halfway. These 5 contacts, we have lengths 28, 40, 55. So that's 27.5. So again, that's going to be 11 for this one too. So even though these are 27, it doesn't pass halfway. And then we have 5 contacts with these lengths. So we got 12, 20, 31, 43, I think. Yeah. 43. So that means we need 21.5. So now this M50 is going to be here 11 as well. So these are all actually the same. So you can see how it's a summary stat, but it's not perfect. And then we have 6 contacts with the lengths 16, 23, giving us 48 as the sum length. So we want the base that passes base 24, which is going to be 12 in this instance. So that's an example how we can calculate these ones. So what we should also note with spades is that you can read up about it and note that we're going to use this command of running spades -- careful with our input FASTQ file and an output directory, that there's also RNA viral spades for RNA viruses. You can see if that makes any difference to the default version above. Then we've got corona spades specifically for coronavirus genomes with the same kind of setup minus the careful parameter. And then we also have a coverage cutoff parameter as well, which can be useful to say add in a value like 10 here to ignore nodes that have very low coverage. But most of our genomes will have coverage that's way in excess of, say, 10-fold coverage. So you can try these different ones. And one thing I would note with our samples is that you might find that these nanomorph samples here, when I ran them, I got most of them decent N50s, whereas for the Illumina data, that didn't look like the case. So you might have to try hard with the Illumina data to actually get it to assemble properly, if at all. You should bear in mind that input data may not assemble well. It may have been amplicon sequenced. So that means there'll be gaps between the different amplified contacts, and therefore we need a reference-guided assembly approach instead of this de novo assembly approach. If you want to scan all your files during this, remember you can use the pattern matching command grep to get the N50 values from a set of directories and the output files and their report files, for example. So note that we also link our work with the WHO protocol for looking at SARS-CoV-2 data, that we looked at, say, using PortShop for demultiplexing and adaptive trimming of nanoprofiles. We used equivalent adaptive trimmings with FastP and the FastX quality trimmer, the same as Tripomatic. We looked at how duplicate reads do occur and how FastP can eliminate them. That was in the previous practical. And then in G03, we'll look at things like mapping tools. And we'll also in G02 look at, well, that's built into the previous steps, but we'll look at G04 on calling mutations as well. So that's kind of how our approach is linked into standard protocols for looking at public health outbreaks. So that kind of concludes this part on the practical. Please see the demonstration videos for actual implementation of this.